I'm going to read from my script. Is it okay?
Sure.
Varenika?
Yeah, it's fine. It's fine.
Okay. So we'll wait for a minute because once we go live, then people can join YouTube link. So Ritu, when you see that at least a good number has joined, then we start. Is that okay? Ritu, can you hear me?
Yes, ma'am. Yes. In a minute, we will go live. We are live now.
Can you see participants? Have they joined? Maybe I take another minute to start.
Yes, participants have joined.
You can mute yourself. IBRC can be mute. Yeah. Okay. Okay, a very good evening, good morning, good afternoon to everyone from all parts of the world. We have a very enthusiastic participation for a much awaited talk. Welcome to the IBRC Lecture Series 2021. We are truly delighted to have Professor Paul Turner from Yale University as our guest speaker today. And this is an endeavor. IBRC Lecture Series is a joint endeavor between Acharya Narendra Dev College, University of Delhi, from where I come from, and Open Health System Laboratory, United States. And the Director of Biomedical Research, OHSL, Dr. Konenika Ray, is also with us today. So may I now introduce Professor Turner to all of you, though he doesn't need much introduction in phage world, but still I need to do my job. Professor Turner is the Rachel Carson Professor of Ecology and Evolutionary Biology at Yale University and Microbiology faculty member at Yale School of Medicine. Dr. Turner obtained a BA in Biology from University of Rochester, a PhD in Microbial Evolution from Michigan State University, and did postdoctoral assignments at National Institute of Health, University of Valencia in Spain, and University of Maryland, College Park before joining Yale in 2001. To Dr. Turner's credit are a large number of highly cited publications, successful case study for phage therapy, and the honors he received include fellowship in the National Academy of Sciences, American Academy of Arts and Sciences, and American Academy of Microbiology. Professor Turner studies evolutionary genetics of viruses, particularly bacteriophages and RNA viruses transmitted by arthropods. Friends, in bacteriophage research, the principal focus of Dr. Turner's lab are of paramount importance to us in the phage community. Briefly, as we all appreciate, elucidating the molecular details of phage binding to their bacterial host is fundamental to understand phage biology, because it is these receptors which confer host specificity and allow phages to differentiate between species or strains of bacteria. So, to identify the phage receptor genes, Professor Turner's group has developed a high throughput tool called transposon insertion sequencing, which makes identification of ideal phages for therapeutic application possible. Another significant aspect of his phage research is phage steering, and their group has validated the promising approach to use phages that not only reduce bacterial pathogen load, but also select for phage resistance mechanisms that trade off with antibiotic resistance. And Professor Turner has also co-founded a clinical stage biotherapeutics company called Felix Biotechnology. And very recently, they have launched this cystic fibrosis phage study at Yale, which is an investigator-initiated single-center trial for targeted phage therapy in the treatment of chronic Pseudomonas aeruginosa infection in cystic fibrosis. So, on behalf of all of us here, I thank you very much, Professor Turner, for taking time out. And many of the registered participants have expressed great pleasure for this opportunity to attend to your lecture. So now, without taking any more time, I invite Professor Turner to give this talk.
Well, thank you so much. That was a wonderful introduction. I appreciate this opportunity and this invitation. And it's just a delight to be with you virtually, if not in person. So let me see if I can get the screen share. Hopefully you can see that. Okay. All right. Well, I'll spend the next 45 to 50 minutes talking about this subject. It is, there's a lot of promise in phage therapy right now. We have a lot of good outcomes. We have still more challenges that we face, especially on this path to making phages generally accepted as a therapy, commercially approved as a therapy. Of course, a lot of this work has been happening for nearly 100 years around the world, but Western medicine is, I would say, catching up to this idea, and it's for an obvious reason. With the antibiotic resistance crisis happening during the background of the current pandemic, but this is something that's not going away. This is something that is destined to be more challenging over time to deal with antibiotic-resistant bacteria. Okay, so let me just briefly introduce my interests. They span three main areas, I guess I could say. On the more basic research side, I've always been interested in how evolution proceeds. What is the tempo and mode of evolution? How do biological systems adapt to challenges? And some images of that are shown on the left. In the middle, this is closer to understanding within pathogens themselves, how do they evolve? What is their ecology? And here we do most of our studies on either directly transmitted RNA viruses or those that are transmitted by arthropod vectors, such as mosquitoes. But the real subject for today would be the applied side of our work. We try to do the basic research that informs applications. And the talk today will consider virotherapy, a form of virotherapy called phage therapy. And the idea is to take evolution thinking to the problem. Some people have called this evolutionary medicine, when you can inform therapy of humans or of any organism really with an eye to understanding the evolution. So that'll be a main focus. And here is the main topic, the antibiotic resistance crisis. This, unfortunately, needs very little introduction. I think more and more of even the lay public really understands that this is a big issue. So there is an increasing proportion of bacteria that show resistance to common antibiotics, and therefore antibiotics simply are not working as well as they used to. The map on the lower left is one of these projection maps for not too distant future from now, a couple of decades from now, where this is expected to be a global problem, and it already is, and it will continue to be. The most populated parts of the world will feel the greatest amount of mortality, but the real message is that all humans on Earth can be impacted by this. To the right is a less common way of discussing the problem, and this is the GDP reduction expected by the same period of time as that map on the left. And it's really showing that in parts of the world where there are countries where already the GDP is low, it's expected to reduce even further, but globally, the GDP would be affected by this issue. A lot of human mortality and morbidity because of the problem. So how would you address this? An old adage is that the enemy of my enemy is my friend, and that is essentially the core idea of phage therapy. Recognized very long ago, as long ago as the co-discoverer of phages, of course, Twort was one, but Felix Durrell was the other. So circa 1917, as long ago as then, Felix Durrell was starting to examine the role of phages and their ability to control bacterial infections in animal models and even in people. He didn't have very many academic appointments in his lifetime. He happened to be appointed at Yale for a few years, but he was very instrumental in the early days of the Eliava Institute there in the upper right, which still exists in Tbilisi, Georgia, has been an amazing place for phage therapy to develop. And if you want to hear more about Felix Durrell, there is a very nice book written about him by my emeritus colleague, Bill Summers, here at Yale University. So really, the reason I'm showing the slide, though, is to remind you of the lytic phage cycle there on the left. And I hope you can see my cursor, but the idea is that phages interact with a bacterial cell that is susceptible to infection. They take over the metabolic machinery of the cell, making copies of the phage, and then those are released through lysis. So this will happen with strictly lytic phages. Some phages have more complicated life cycles than this, and I won't really focus on them today. But the immediate thing you can see with this process is this is somewhat similar to a self-amplifying drug. So if you have a minimal dose that is applied, you can be very efficient in terms of treating an infection. So that's one of the benefits. There are two benefits that I note in the slide. The specificity of the phage to its target means that you have a drug that's specifically acting, and it can self-amplify to make copies of itself, as I just mentioned. But there are obvious costs. There can be limited spectrum because of the specificity. It could be difficulties in delivering to the infection site in the human body, and there could be immune recognition over time. Even though you ingest phages every day, probably you breathe them in, attached to dust particles, you drink them by accident and eat them by accident. So you don't mount a big immune response to them anyway. And yet that would be different potentially if you took 10 to the 10 phages every day for 10 days. I would think that there could be this possibility of your immune system catching on and you would be unable to receive that phage treatment in the future. But as an evolutionary biologist, what I'll focus on for the next few slides is that we would expect bacteria to evolve resistance to phages if they're used in therapy. And that's because bacteria are quite good at evolving resistance to phages altogether. Here are a couple of images that remind of this from a recent review. So on the left, you could think of the defense mechanisms that bacteria carry around with them all the time. They can be modified through evolution, but a lot of these mechanisms would prevent the phage from attaching to the cell exterior because the binding mechanism is altered. I'll focus a lot on that today. But even consider after the fact, if the phage is able to bind and the nucleic acid enters, it could be prevented from going to the completion of the replication cycle. That's essentially the details shown here on the left. You can read this review for more explanation of it. But I wanted to quickly touch on the more recently understood and discovered mechanisms, CRISPR-Cas on the right. So here, there's a form of an adaptive immunity. It's quite analogous to adaptive immunity that you see in multicellular systems, in the sense that a bacterial population through time can be more resistant to certain phages that are attacking because they build up an immunity in what's called a CRISPR array, something that is modified over time, allowing them to become resistant to phages that would otherwise take over that cell and try to kill it. So these are pretty amazing mechanisms and quite a variety of defense mechanisms that bacteria have and that can further evolve. So I want to take just a couple of minutes to remind the audience a bit more about evolution and how it proceeds. And this is going to set up kind of something that we see all the time in evolution that we're trying to exploit in phage therapy, but I want to go through this a little bit carefully. So recall that Darwin had amazing insights in his career. Charles Darwin, in his most famous book, Origin of Species, he talked about natural selection. Even though others tried to articulate similar processes before, he was the one who is given credit for understanding it the best. And he articulated that if you have individuals in a biological population and that they vary from one another, if that variation is heritable, then some of those variants are going to be advantaged in survival and reproduction. So over time, the population can evolve to reflect the successful variants. So those are the steps by which natural selection proceeds. The traits that evolve and that make organisms better fit in their environment relative to organisms that lack that same trait, these are called adaptations. And this is a very beautiful idea for which there is abundant evidence that it's an actual, it's very common in biological systems. And interestingly, if you found life on other planets, if some of these upstream things still held, individuals in the population varying and the variation being heritable, then you can think of natural selection as one of the few universal forces that could be existing in biology. In other words, any population with variation in trait inheritance should be able to go through this process. But there is something that comes along quite frequently with evolution, and that's called trade-offs. So as evolution proceeds, it can lead to compromises. And this is the process of natural selection improving one trait, but not simultaneously improving all traits. And in fact, sometimes traits improve at the expense of reduced performance in other traits. So there's lots of ways for me to illustrate this. I often talk about it in terms of these two familiar carnivores. The cheetah on the left is evolved for speed. It can chase down fast prey, but if it tried to bring down a slower moving but larger prey item, it would be unable to do so because it is just not a big enough predator. And essentially the lion is the opposite. It is a big lumbering carnivore that is able to jump on the back of a zebra and bring it down for a kill, but it is not fast enough to, say, chase down a gazelle as a prey item. So these carnivores, the point of this slide is that the carnivores depicted here are doing very well in their environment, but you can think of them as not exploiting all aspects of their environment, and this is due to compromises. So now I'm going to explain an example of this for phage bacteria interactions, and it's really the system that I first started studying this problem way back when I was a postdoc with Lynn Chow at University of Maryland, College Park. And what I'm going to show in the next few slides for a plant pathogenic bacterium in this phage called phage, what we observe there extends to informing how we handle phage therapy now through our research. So I'll get there in a moment. Here's a cartoon prediction of phage phage on the left, and this kind of washed out image in the middle. You can see these small white circles. These are phage particles that are attached to the type 4 pilus extending from this bacterial cell. And that bacterial cell happens to be Pseudomonas syringae, which is a very common plant pathogen. Here on the right, you could see cells of Pseudomonas syringae moving across a leaf surface and entering through the stomata. They do this through what's called twitching motility. And the type 4 pili allow them to move across the leaf surface in this way. So I'll now illustrate a problem of this interaction in terms of the bacteria being able to maintain their ability to serve as a plant pathogen. So I'll remind you that here's another image of the phages attached to bacterial pili, but what I'm really highlighting are the two images on the right, where these bacteria are important in a form of plant disease of legumes, and it's called halo blight disease, and they can enter the plant and cause this infection. But if you consider how can the bacteria escape phage attack if the phage are in their environment and there is selection pressure, they can simply get rid of the type 4 pili because that is the binding target. But this leads to an immediate trade-off. If they lose the pilus, then it decreases what's called their conditional virulence. That is, the bacteria will no longer be able to traverse the leaf surface and enter through the stomata, and therefore they can't cause the disease through the ordinary process. It's shown in laboratory studies, highlighted here in the lower left, that if you take the type 4 pili, if they lose the type 4 pili and you physically place them inside of a plant as a researcher, they can infect the plant. But the point is, naturally, without those type 4 pili, they can't traverse the leaves, although they could still be infectious if they were purposely placed inside the plant. That's a conditional virulence factor. But the main message here is that phages can select for reduced bacterial pathogenicity. And this is something that I've been thinking about for a long period of time, as I said, in these phage experiments when I was a postdoc. But seizing on that idea, the idea is can phage therapy exploit these sorts of evolutionary trade-offs to our advantage? And this is where we've done a lot of work in the last few years. This image from one of our reviews in 2019, I'll quickly walk you through how this can happen through a switch in antibiotic resistance to become sensitivity and a switch in virulence to become low virulence in the system. But hypothetically, imagine the cell, which has some characteristic traits. First, focus on these orange and red structures around the cell. These are efflux pumps. We're trying to illustrate efflux pumps here, which are protein complexes that some bacteria have. And if antibiotics get inside of the cell, these protein complexes will actively pump them out. So this can create multi or even pan-drug resistance to a wide variety of antibiotics in some bacteria. What can happen is that if a phage is interacting with the outermost protein of that efflux pump, then this is the way that as a lytic phage, it can bind, enter, and kill. So therefore that bacterial population will undergo selection from phage attack, and you'd expect it to evolve resistance. One way to do that is to alter or even eliminate these efflux pumps, despite the fact that they have multiple functions. And if that occurred, then the bacteria would switch from antibiotic resistance to antibiotic sensitivity. In other words, you could use phage to kind of drive bacterial populations back to an antibiotic sensitive state instead of the antibiotic resistance crisis that we're dealing with. A different example, I'll go through it quickly because I'm not really talking about virulence very much today, is imagine a purple capsule surrounding this bacterial cell that shields it from detection from the immune system cells in your body. A phage could be using that as a binding target, the capsule, and again, this will kill the bacteria, placing selection pressure on the bacterial population to escape phage attack. They could do that by altering or even removing the capsule altogether, but then they switch from becoming high virulent to low virulent, and they become less important biomedically. So in this way, we're trying to find phages that force bacterial populations into a compromise. They will be used to kill them in the classic sense of phage therapy, but they can also be used to steer them in a better direction for biomedical applications of antibiotics, especially. So our premise in all of this is that we should be able to find such phages, that we wouldn't even need to engineer them. It is possible potentially to engineer them, but the point of this cartoon, and we mentioned it in our review, is if you look hard enough in the bacterial world and the phage world especially, you should be able to find such phages that have naturally evolved, and you might be able to exploit them in therapy. So I want to now go through the steps that we undertake and what could be considered a pipeline of discovery and ultimate placement into emergency therapy cases. These are personalized treatment cases of individuals suffering with multi-drug resistant bacterial infections. And as was mentioned in the nice introduction, we're now at the point where this technology has advanced enough that we can put it into investigator-initiated clinical trials. That's very promising, because that is definitely the next step that would be needed in the Western world, or at least certainly in the United States, to take this into something that is a generally approved therapy. But here, let's start with the first step. Where do you find phages? And essentially, I can tell you, you can find them everywhere. So you can look in a more or less pristine environment like this lake nearby to my home in Connecticut. You could look at very human-altered environments, like this member of my lab team, Ben Chan, as he's sampling in human-altered environments in Haiti and Democratic Republic of Congo. In the upper right here, you've got a sewage treatment plant, which classically, in the relatively modern era, people can even take school groups there to sample water that ultimately can lead to phage isolation in the laboratory. And a place where you can find a lot of antibiotic-resistant bacteria is in animal agriculture. So that might be a place where you'd also look for phages that are exploiting and trying to kill antibiotic-resistant bacteria. So phage hunting is very possible. Once you get your phages, if you can afford to do this, it's good to subject them to whole genome sequencing, so you know what you're dealing with. This would reveal whether the phage is carrying around any genes that actually might be harmful to humans. It is possible for phages to carry around toxin genes, for example, and that these, upon lytic infection of hosts, they might integrate in to host bacterial genomes, making them more toxic. That's just one example of something that's plausible. It's not necessarily you'd see it all the time. The point is, through whole genome sequencing, you could look for such dangerous genes in a library of phages that you might be considering for therapy. So this somewhat busy cartoon slide is depicting a recent library acquisition of phages that are specific for Pseudomonas aeruginosa, and they're all strictly lytic. And you can see how we know their genome sequences and relatedness to one another, so we can place them in different known phage families. Therefore, we have some understanding of them based on close relatives within those families. And I'm not really showing it here, but one thing we are avoiding are finding phages that have complex life cycles, like temperate life cycles, where they would force the bacteria to be a lysogen. And that means the phage would hang out in the cytoplasm or integrate it into the genome of the bacteria, and it would be inherited across generations of the bacteria by daughter cells, not necessarily killing the bacterial cell through lysis. It doesn't mean that those phages couldn't be used in therapy, but they have a more complex life cycle that's a less efficient way of killing the bacterial cells, and we are, at least for now, avoiding them. So the point is, so far we've got phage discovery is straightforward, and you've got isolation that can be used with genome sequencing. But now it's very important to think about, are phages so specific that they wouldn't be useful? And I think a lot of that understanding is basically an assumption from some well-studied phages of the last century. And what we are finding, and others are finding, is that not necessarily is that the case. You can find phages that are naturally occurring that cover a lot of genotype space in the target bacteria. Now, this slide is kind of a typical matrix of, along the left, you've got candidate phages and an isolation library from the wild, and across the top, you've got various types of bacteria that I won't give you the details of them. Here in red would be where there's a complete mismatch, but some of these are controls. If you have a phage that is, in this case, able to infect Shigella bacteria, which causes a form of dysentery, which is very harmful to humans and can lead to especially childhood mortality, we can find certain phages straight from nature. There's a lot of green here where they are infecting various types of Shigella bacteria, whereas in red, they cannot infect other species of bacteria. That's what we expect. Here's another matrix where, again, aligned on the left, we have some candidate phages. In this case, they're infecting Pseudomonas aeruginosa. And here we're using challenge bacteria across the top that come straight from the clinic. These are from lung infections of human patients. So we'll do whole genome sequencing on those bacteria, and we'll fill in this matrix with the dark blue squares indicating when this phage can kill the challenge bacterium across the top. And you'll see that there's a lot of dark blue in this image, meaning that you can find phages that are naturally killing a lot of these clinical isolates. Trying to make sense of the killing is difficult. So if you look at this phylogeny, there's not a whole lot of immediate patterns to that phylogeny explaining the outcomes of the matrix. So this unpublished data set by Ben Chan, a research scientist in my group, a senior postdoc, this is the kind of work that we're trying to do to challenge bacterial isolates, whether certain phages are good at killing them, and ultimately it's going to inform which of these could we use in therapy. And that's where a lot of further research comes in that I won't talk about today. Happy to talk about it in the Q&A, but there could be cocktails made up of specialist phages, or you could have cocktails made up of more generalized phages, or you might even be able to find a phage that is so good at broadly killing genotypes of the target bacteria that you just use one alone. So there's lots of ways to do this. What I want to focus on for the next few slides is a very important problem, though. Everything that I've said to date here in the talk is that you're going through this pipeline of discovery, but our hope is that we're going to find phages that interact with certain bacterial receptors, especially those receptors that are important in antibiotic resistance, for example. And you want to do this so that you can find a phage that is exerting this trade-off that can be exploited in biomedicine. But first you have to figure out what is it attaching to in the first place. And here I should reveal that this is very important work in virology. As we've seen with the current pandemic, lots of viruses that exploit humans and domesticated plants and animals, but also in phages. It's not trivial to figure out what phages or viruses are binding to. So we've developed a relatively inexpensive technology because it harnesses existing in-seq technology, or transposon mutation insertion. You can read about this in a paper we published last year in Proceedings National Academy of Science, but the cartoons in the next few slides and the images, I'll walk you through some of these results. So what do you do here? You want to take bacteria and subject them to transposon insertion that creates a mutant library. And what you can do with that mutant library, these are single gene knockouts in your library, and you're going to subject it to something that is going to select for those mutants. And in our case, we care about phage selection. And this will enrich in your library for the bacteria that can best avoid phage attack because their genes have been knocked out for which those phages rely on for binding. And the image here in the lower left is a cartoon of what the output data are going to look like that I'll show you in a moment. The genes for phage infection would be in the upper left quadrant here in blue. And that's what we're looking for in these enrichments, where you're looking at the output counts per million bin by gene versus the input. Now, the first thing you want to know in your transposon mutant library is whether you've got good coverage around the entire genome. Otherwise, you're going to miss a lot of potential genes that are important for phage binding. And this is an image directly from the paper, and you could look to it for details, but it's essentially showing you the entire E. coli genome that was used in this study and highlighting that there might be some regions of over amplification and representation in the library, but the point is that you've got complete coverage around the whole genome, and that is what we would like to see. With that coverage, and then you look at the output data, we included in the study some controls. So let's begin with this image in the middle. This is phage T6. It's already known that this well-characterized phage uses the TSX porin of E. coli as its binding site. So these output data, the best hit, the most statistically significant hit is TSX, and that's quite satisfying because that is a control that worked. It confirmed the known binding site that's been described for some period of time. Here on the right is an example of a phage that's essentially straight from nature, and we know that it is lytic and can kill E. coli, but we don't know much else than that other than its full genome and the family it resides in. It happens in this output that in this particular case, TSX was also seen as a binding site for this poorly characterized phage that we put through this process. Now, that doesn't mean all phages will bind to TSX. It's just happened to be the case in this example. If you look at the paper, we describe 6 total phages that are straight from nature, similar to U115. And we also describe the statistically significant output in this analysis to tell us, as in the case of U115, what it is binding to. So again, a reminder here is this particular phage, I'll give you a bit more data on it. It uses the nucleoside porin, TSX as a receptor. Now, this kind of work needs additional assays for confirmation. So if we suspect that TSX is the binding site, you can then take the wild type bacteria and lots of knockout engineered strains that you created in the laboratory. You could look at other suspected binding sites and eliminate them as important. But this upper graph is essentially showing you that this U115 phage cannot be detected for its plaquing if you knock out TSX. If you put in a rescue plasmid that is still knocked out for TSX, you don't get any binding there, but you start to get some growth of the phage when you rescue the bacteria by putting the binding site back in. In the lower right, this is something I want to key in on. It's a classic adsorption assay where you're looking over time at different bacterial strains and you're characterizing how well did the phage bind to these strains over time, and you're expecting them to disappear from solution if they are binding. So here in the case of the black line, it's showing you that indeed, when the TSX protein is there made by the gene, then you get the expected binding of the phage and it disappears from solution. When you eliminate that binding target, the phages just sit there in solution and they're not binding. So these types of assays are critical because it can give you even more confirmation and confidence that what you're observing is true. Now, if you have all that in hand, we published a paper last year. I believe it's already available, Current Biology, even though I'm saying in press. But you can take a look at that paper, and the lead author was a current postdoc in my group, a research scientist, Alita Burmeister. So what we described, and I'm showing in this cartoon image, is there are lots of ways that if you find the right phage, depending on what it's binding to, you can force bacteria through this process into becoming more antibiotic sensitive. I walked you through earlier the interaction of phages with efflux pumps can lead to killing of the bacteria and selection for the bacteria that are becoming resistant to phage attack, but then are suddenly vulnerable to antibiotics. In the middle is the another example, which can again involve here the capsule, where I'm highlighting that capsule changes in the bacteria due to phage selection can also be important in easier entry of antibiotics into the cell. And essentially the same analogously could happen with what's called lipopolysaccharide. A lot of these around the bacterial cell are giving it integrity and structure. And these are used by phages in binding quite frequently. But also the depth of the LPS on the exterior of the cell can be something that can help shield it from antibiotic entry. So as you kind of shave away that LPS due to phage selection, it can make it easier for the antibiotics to enter. So all these would be great outcomes for phage therapy because you can synergize phages with currently useless antibiotics and make them more useful. So the best characterized system of this, again, we showed it in an E. coli model, and we had a paper on that last year that you might want to take a look at. And this is, again, work led by Alita Burmeister in my group, but this is phage U136B. And this rather busy slide here on the left is a structural diagram of the Toll-C efflux pump in E. coli, where here at the top is the outermost portion of the Toll-C efflux pump where the protein Toll-C is exposed to the environment, but this protein complex, as I said earlier, can be responsible for actively pumping antibiotics out of the cell. It has other functions also shown here in the lower left. Now, in the middle, if you find a phage like U136B, we've done, again here, knockout challenges where the ability of the phage to plaque here on the left axis across different types of bacteria with different knocked out genes. In the case of, if you remove TOL-C, you can't get any visible plaquing of this phage. It is highly reliant on TOL-C being present for binding and entering, whereas you can knock out other genes and it seems to be growing just fine on the E. coli bacteria. But the real reason to highlight this is the problem that the remaining bacterial population, which are mutants that avoid phage attack, how do they change in their tetracycline resistance? First, in a control where you grow the wild type bacteria, an engineered TOL-C knockout, and a spontaneous phage resistant mutant to this phage, they all grow normally and pretty much the same in an antibiotic free environment. Whereas if you put antibiotic into the environment at a particular level, then suddenly the TOL-C knockout, as expected, cannot grow as well because it cannot actively pump out the tetracycline. And the same is true for this phage-resistant mutant. And that is exactly what we would love to see in terms of a phage driving a trade-off. Here is another example in the same phage where it's interacting with a co-receptor of LPS. I'm showing you a side view of LPS here on the left, and the colored portions are where the phage is binding at a particular depth of LPS. And what that will do is if you remove those genes and knockouts, then the phage fails to bind, and you can show that in these failed growth of the phage to be able to grow on those types of bacterial knockouts. But here on the right, the interesting consequence is that the phage-resistant mutants, they avoid phage attack, but due to the shaved down LPS, they become more sensitive to the entry and are sensitive to colistin, a different antibiotic. So again, you've got an entirely different method here where the phage can be used to steer the bacteria to greater sensitivity. Now, the complexity in all this is that we can't control it in the human body during therapy. So we've done some assays that speak to that, and they remind you that here, let's begin with this graph here in the middle. If you look at a collection of these spontaneous phage-resistant bacterial mutants, and you look at how resistant are they to tetracycline if they undergo changes in efflux pump genes, then many of them don't change their sensitivity to that drug. Many of them do, and that's what you would love to see through a trade-off. But alarmingly, some of them have the ability to resist the phage, and they'll have a slight increase in resistance to tetracycline. So there are lots of different genetic outcomes that are possible. If they were changing LPS, the overall outcome is better in biomedicine, because here, pretty much they keep the ancestral wild type level of resistance to colistin, but the vast majority of them are now more sensitive to colistin. So again, these are the genetic possibilities, and I'm showing you that they're quite complex in terms of how the bacteria can solve the problem, sometimes leading to trade-offs, unfortunately, sometimes leading to trade-ups. And a trade-up is definitely something you want to avoid in this kind of therapy. This would not benefit it. So I'll end with this little portion before going on to clinical evidence to show on the far right here, if again, phage and antibiotic, they're both trying to enter the cell, if they're trying to enter through the same exact portal, then you really have a high likelihood of a trade up. So as the bacteria evolve resistance to the phage, now they're going to evolve resistance to the antibiotic because they're coming in at the exact same entry point. And we have evidence of this in an unpublished data set. You can see some details in published work already for an antibiotic called albacidin, which is known to enter through the TSX porin and two phages, one well-characterized T6, another one that I mentioned earlier as a recently isolated phage of E. coli U115. So if they're all entering here through the same mechanism on the left, this porin, you'd expect that if you independently obtain mutants to all three types of antibacterials, you'll probably get evolution of cross-resistance, meaning that they become resistant to the phage and the antibiotic at the same time. That's exactly what happens as revealed through whole genome sequencing. We find that different classes of mutations can be responsible if you look at collections of these mutants and how they underwent the mutation. But here in the lower right, if you look at the TSX gene itself and where these mutations are happening, in all of the mutants, this is where we see a hotspot for the change. So again, it's a quick reminder that this is a very promising idea, but a lot of basic research should be done to think of exactly, how good would your candidate phage be? And would it be on average good enough for therapy, or would it actually be potentially problematic? So now let's turn at the end of the talk to some clinical evidence, because this is really where we have some confidence moving forward, despite these complexities that I just explained. Phage discovery on the left here, showing Ben Chan in the environment, isolating phages, can lead to immediate characterization of some promising phage candidates. And even before one does abundant animal studies, you may find desperate people who need therapy. And in the case of this young woman in the state of Texas here in the United States, she's being shown a vial of phages that came straight from the environment, but we made a clean prep of them that she's going to inhale into her lungs to try and have those phages kill a chronic infection caused by pan-drug resistant Pseudomonas aeruginosa in her lungs as her physician is looking on. So this kind of work is very promising. Others have talked about it in terms of emergency phage therapy helping individuals. Our one published case study, I won't go into it in detail, I'll just highlight it briefly here with some cited work in case you want to learn more. The first approved study that we did was a man who was elderly and he had an aortic arch replacement, which is not an uncommon surgery, but it is a significant one because it's putting in an artificial part of your heart. And unfortunately, that substrate can be an easy one for bacteria to come in and cause a chronic infection on that artificial substrate. In his case, he had a chronic Pseudomonas aeruginosa infection that was multi-drug resistant, and he was too elderly to undergo through the surgery to replace that infested piece. So we used a phage that we found in nature, synergized with an antibiotic that was useless in his treatment, and we synergized to make it useful by killing the bacteria, enriching for mutants that were sensitive to this antibiotic, and that helped clear the infection entirely. So this kind of good outcome is highlighted in magazines such as Scientific American. They talked about this case and some other ones as promising in Western medicine. We've been slower to publish on these other great outcomes. And here, 12 out of these 13 total cases, you'll see that they are mostly people with lung infection issues because of what's cartoon depicted on the left. People with cystic fibrosis have a genetic disease which causes them essentially to have a different lung architecture than other humans, and they'll have widened lung airways with overproduction of mucus, and this can be a great substrate for bacteria to come in, take hold, and cause a chronic infection. And as they are increasingly multi-drug and pan-drug resistant, this is a huge biomedical problem because it can lead to lung failure and death. Non-CF patients also can suffer through this if they have bronchiectasis or chronic obstructive pulmonary disease. And very, very many people have these problems, and this is what is alarming with the rise of antibiotic-resistant bacteria, is you're going to see more and more increased morbidity and mortality. So this is a quick table showing you over the course of about a 2-year span, the very many people of different age groups and genders that we helped overcome lung failure, and we're looking at death even in the case of chronic infection. And we were able to use phage therapy through inhalation, delivering the phages to their lungs. And we're excited to keep publishing on this work. A quick snapshot of the kind of data that we have that are unpublished, but I've been sharing publicly so far, is that consider these patients who are inhaling the therapy, they can also give us sputum samples that they cough up of lung mucus through time. And we can look at their sputum samples for the bacteria and the phages or anything else that's present pre, during, and post therapy. Some things we would want to track are shown here on the right. Do they undergo improvement in their lung function? And you can measure this through the amount of air that they force out of their lungs in a single second. And the kind of data that I'm showing here in the middle are supportive of this working, where after phage treatment, their lung infection improves. And at the same time, if you're showing that their bacterial load decreases, then that correlates to the therapy being very helpful to them in driving out the infection, or at least reducing it to a great deal, allowing them to have better lung function. And then the lower right is the support for the trade-off working in vivo as it worked in vitro. That is, you can have these patients flipping from antibiotic resistance to sensitivity, and that's wonderful for the bacteria that you can't eliminate. At least you can better control them if they become problematic. Now, all of this is prediction, though. So we're making evolutionary predictions for these trade-offs. We don't exactly know how the in vitro data will predict the patient outcomes, and I'm admitting that this is still somewhat operating in a black box, and we're trying to fill in that black box with more details. Phage therapy itself can be predicted to work, and we've shown that to be true. but there's lots of basic research that needs to examine how phages that enter your body through therapy, they can be interacting with your own cells, even if they're not replicating in them. They could be interacting with other phages in your virome and prophages, even in the target bacteria that you're trying to eliminate. There could be traits changing through evolution in response to these phages that are different than the ones that you predicted, and you would want to know what those are. You would like to know what's happening with other bacterial pathogens that could be in the same target site, and are these non-target bacterial pathogens going to be somehow competitively superior because you're removing some pathogen that's competing against them? And non-target pathogens, especially viruses and fungi, could be in the human body, and you would like to know how they are either hurt or not by the application of this therapy. So in a sense, all of these could be examples of what's happening in your microbiome, and already microbiome research is very complex and yet exciting, and as we layer in phage therapy, we need to know how those interactions really work. Now, I want to end by, in a weird way, relating what I've said all along to another side of my research that I didn't talk about today. Now, as we grapple with the pandemic and try and get this under control, then this is essentially, of course, an emergence problem. And humans are facing pathogen emergence, especially RNA viruses, the coronavirus, SARS coronavirus 2, and prior coronaviruses. We have a lot of understanding from HIV, which emerged in humans decades ago. Influenza virus is something that we always see, but new variants can emerge. And we want to better predict how this happens. Why are some viruses good at doing this? And are generalist viruses better to emerge on, are they better poised to emerge on humans in the future? So that's some of the other work that I do. And in a sense, you can take those questions and apply them to phage therapy. The goal here is to better predict how the phage potential is to broadly kill the bacteria. And why are some phages good at infecting lots of bacterial strains, whereas others are not? We would like to exploit those generalist phages and know, however, whether they're going to emerge on microbiome species. So my point in these last two slides was to show that a lot of the work that somebody can do in other aspects of virology can actually carry over to the helpful ability of viruses through virotherapy, and better understanding both the bad side of viruses emerging as pathogens, as well as the good side of controlling and understanding how generalist viruses can work in our favor, and virotherapy. These go hand in hand. As you do this kind of work, I spent some time today showing you clinical evidence from Pseudomonas aeruginosa. This is a slide from our startup company, Felix Biotechnology, reminding that we already have evidence through our unpublished work of phages that exert selective trade-offs in other target bacteria. I didn't have time to talk about them today, but you can think of this work in our laboratory and those of others around the world as advancing this through discovery to a preclinical phase of personalized medicine and ultimately to clinical trials. And that's where I'm going to end with a couple of slides on next steps, highlighting that we are delighted that we have obtained the resources to do our first clinical trial on the prophylactic ability of phages to protect the CF lung, the cystic fibrosis lung. We are teaching this technology to students at Yale University through what's called course-based undergraduate research. This is where they enroll in a course, but they essentially learn about phage discovery, genomics, and applications. And we're also doing this work in workshops around the world. So this happens to be an image from our first West African phage workshop, where we were able to teach the technology to physicians and researchers there. A reminder that the antibiotic resistance problem is beyond human systems, and this is threatening food security. So in plant and animal agriculture, we're quite worried about the rise of antibiotic resistance there, and also in aquaculture. So the idea is, analogously, could you exploit phage therapy to help those situations? Also in zoos and aquaria, already members of my lab team, they were involved in helping a sea turtle in Florida, here in the United States, that had a multi-drug resistant bacterial infection, and we isolated phages to help treat that animal and save it from succumbing to that bacterial infection. So the last thing is, I start to get this, maybe some of you are thinking of this following question, I'll end with it. If you used phages in a widespread way, and is there a concern that this would select for broad resistance in bacteria, causing a problem that's analogous to the current antibiotic resistance crisis. In other words, if you use broad-scale phage therapy, would you kind of recreate a problem in the future that we are grappling with today for antibiotics? And I think this is unlikely. And we have a paper that was published last year. You can go take a look at it in Virus Evolution. And it's unlikely because the mechanisms by which bacteria share antibiotic resistance genes through horizontal gene transfer, especially the movement of conjugative plasmids and the easy ability to obtain resistance genes to lots of different antibiotics, is completely different than how bacteria might get through horizontal gene transfer the resistance to phages. It's going to be on average less useful, let's say, for them to obtain a CRISPR array from some other bacterium genotype or even a species, because the phage resistance that they would acquire wouldn't necessarily be useful to them. Similarly, the broad spectrum antibiotics that we use all the time, these exert general selection on bacterial species, lots of them, to obtain antibiotic resistance. But this is going to be unlikely. If you deploy a generalist phage, it's generalist in the case of covering a lot of genotype space in the target bacteria, but it is very unlikely to transcend the ability to infect other bacterial species. So again, you can look at some of these musings on why we don't think we would be recreating a current problem. I'll end with the acknowledgements to the folks who are currently and past members of my lab team today. In the upper right here, I talked about Ben Chan's work, Katie Courtwright, a grad student in my group, as well as Alita Burmeister. Also, our physician colleagues, John Koff and Gail Stanley, I highlighted their work. I'll end, since we're running short on time, I'll just end with briefly thanking my other collaborators, members of our startup company, Felix Biotechnology, and our generous funders. Should I stop sharing and then we'll, is that your preference? Okay,
Yeah, that'll be
we'll
okay.
do that.
Yeah.
Okay.
Thank you so
All
much.
right.
Thank you very much, Professor Turner. That was a lot to learn in this 45 minutes. Yes.
Thank you.
And, yeah. And yeah, as you rightly said, that the basic biology, there are a lot of gaps for us to understand before we take phage therapy. And your work is both in terms of therapy as well as understanding the basics, like especially finding the genes for binding and then learning the evolutionary trade-offs. So this is a lot to learn, and we have several questions. But before that,
Oh,
would
great.
you like to, like you have this Felix Biotechnology company where you're going for clinical trials. So
Yes.
would you like to share a bit about the regulatory questions you were asked before you go ahead with this?
Oh, yes. Yeah, there were very many. So the U.S. Food and Drug Administration is supportive of our work, and I thank them for helping us with our personalized medicine cases, emergency treatment that we've done. But it is a much bigger deal to put this into a clinical trial and to use healthy volunteers to examine the problem. So the big issue is manufacturing. How are you going to make a phage stock that is the same in large volumes and to have this constancy across your phage doses? So here, what we had to do was work with a company, and we worked with Advanced APT, I should say, I'll just give them by their abbreviation, a company in Gaithersburg, Maryland, and they have a facility that is able to manufacture the phages for us. So here I'm highlighting that the manufacturing process, the demonstration that that is completely clean, that you're not going to be having any additives in there from cellular debris, et cetera, that could be physiologically harmful to humans. You need to make sure that you are producing a product here that is constant, it is safe for people to be administered. So I would say that's a big regulatory hurdle that we have to keep in mind. The other is, how are you going to design this trial? And what we did, I'll just go through it briefly, is that we wanted to design a trial that looks at the prophylaxis, because ultimately that would be the end goal. That would be terrific. If you give people a phage or collection of phages that protect them from the entry of bacteria taking hold and causing a chronic infection, especially in cystic fibrosis patients, this is what you want to achieve. So to be able to do this, you have to look at people over a multi-month period when they might be naturally experiencing these infections, and showing that the group that have the phages are better protected than those who are in a placebo group. Now, the last thing I want to say is it's ideal that you don't want to hold back this new promising technology from anybody. So therefore, you want to do what's called a crossover. So the individuals don't know it, but they'll be in the placebo group, and then they'll move over into the treatment group and vice versa. So in this way, we're examining with each person, how do they do in the time period for which the prophylaxis is there versus a mock prophylaxis or a placebo. So I hope that those two kind of highlights are indicative of the things that one needs to take care of here, but I'm happy to provide more details if you prefer.
No, that's perfect, because the prophylactic uses of phages is very much needed for the frontline workers also, like the doctors and nurses who attend to patients with high infection, lung infection. So if phages are demonstrated to be good prophylactically, then it will be having wider applications, not just for cystic fibrosis patients.
Very true. I completely agree. Yes.
So with your permission, now should I take questions from the audience? We have
Sure.
collected. Okay, so we have several of them. I may start with Shinto James. Shinto James wants to ask, what kind of evolutionary forces drive certain phages to evolve specialization to a narrow host range?
That's great. So I think the rules by which phages are either highly specific versus general, of course, that's a general, that is a bigger question. In all of virology, we want to know this, as I kind of touched on for predicting pathogen emergence. So I will quickly say one plausible scenario is if phage is in an environment where only one particular type of bacterial, either species, but especially genotype, is very prevalent, then we consider this in evolutionary biology kind of a luxury. So if a biological system is challenged by kind of one target, then it can be quite specialized in becoming adapted to be good at thriving in that environment. So it could be through time, as this proceeds, specialization kind of narrows opportunities sometimes. The genome will be so concerned with that one selective target that essentially it doesn't have to worry about other selective factors. And it might even kind of strip away or just lose through genetic drift some genes or portions of genes that might be useful if that environment changed. But the consequence is that if the environment is not changing very much, then specialization can evolve, and it leaves a biological system pretty narrowly capable of exploiting an environment. And that's quite possible in virology. We don't know the exact rules for that, but even in viruses of humans, you see that often. A quick example would be variola virus was eliminated through a vaccine campaign, and that virus that causes smallpox was more easily eliminated because it has no known animal reservoirs. It uses only humans. It was selected to use only humans. And when you had a massive vaccine campaign around the world to remove the virus from its natural environment, it is no longer circulating as a natural pathogen of humans. So the same analogy can occur in phages. We don't know the exact details, but the same analogy can occur.
Right. Next question is, Dr. Turner, in your studies on bacteria evolution, this is from Woon Kit Chin. And have you seen certain hotspots where mutations occur that drive resistance to phages? Do you think it's possible to predict these locations?
That's an excellent question, because these are the kinds of data sets that we are grappling with now. I didn't touch on that very much today, but think of it in the sense of, as the person who's asking the question articulated, if you have a very large number of phage-resistant mutants, can you look for SNPs? Can you look for hotspots that govern that resistance? Even though you're expecting myriad possible genetic solutions, that doesn't mean they're going to happen all across the genome. They could be happening in places that let you better understand genetically what is occurring. That's definitely what we're seeing. So the satisfying result is if you see in genes that are responsible for the receptor that the phages are binding to, there might be several genes, several mutations in that gene for the bacteria that are happening, but they're kind of confined to that region. The problem is that's not the only thing you see. Sometimes you see new mechanisms must be at work that are being altered through genetic changes elsewhere that you didn't expect. And one way for me to sort of vaguely explain why that would be is a gene network. So these are studied in bacteria and other systems. They're easier to know in bacteria because the genome sizes are smaller. But this is where you have traits where there are several genes underlying in a network talking to one another, if you will, to produce that trait. And there could be mutations at many places in the network that you don't predict or understand that could give the bacteria resistance to the phage because it's all part of a network. So I'm not saying that that kind of genetic complexity is not solvable. It is. I'm just saying that it needs more work. It's in the realm of the basic research in bacterial genetics that follows along with the promising phage therapy that we and others are doing. You know, that's remarkable time and energy that would be well spent, that people could understand this better. And I think it's going to lead to a better understanding through necessity of gene networks, in especially some bacteria that have not been well researched for that problem, but they're very important for antibiotic resistance pathogens causing infections.
Right. Next question is from Varun Babra. Because this is the mode is such that the audience are writing, so we can't have a follow-up question. So I will end at that and I'll take the next question. So this is Varun Babra from Mysore Bhopal, and he wants to know that can we perform phage-directed evolutionary trade-off to make pathogenic bacteria become sensitive to antibiotics?
Mm-hmm. Yes, so I think it's very promising that we see this not only in vitro. The question is, if you're doing this in the body of a human or many humans, are you really getting that same response? We believe so. We have unpublished data that show that the in vivo case is the same as the in vitro prediction. And I think the question is talking about how generally promising would this be to kind of push back on the antibiotic resistance problem. And that would only occur if you were using this very, very broadly. And of course, that's what we're trying to get the confidence so that physicians around the world could be deploying phages against particular problems. And I would expect if you're doing that at a broad enough, large enough scale, you would maybe see these trends back in the promising direction of reduced antibiotic resistance and greater antibiotic sensitivity in systems that we care about for human opportunistic bacterial infections.
Right. Thank you. So I'll take two questions now together. One is from Ujjal Kumar, and he wants to know that have you observed any kind of phage interactions with viruses? I mean, animal viruses, I think he wants to ask. Right. And the other question is not related, but a very oft asked question, that what kind of immune response one anticipates when phages are administered for therapy?
Right. Great questions dealing with human physiology. So the first one is, we've never observed any direct interactions between phages and viruses of humans. And I'm not saying they can't occur, but we've never observed that. And it's not as if the literature is abundant with anything about that. But it is important to know, and we have some unpublished data with Jennifer Bomberger's group at University of Pittsburgh, that, and others have seen this in the literature, what I'm about to say, is that phages can be interacting with your cells, even entering your cells, and even eliciting an interferon response from your cells without replicating in your cells. So why do I say this in response to this question is that the production of interferon is something that is important for viruses that might be trying to infect a human that is in the human body alongside a phage that is eliciting this response because interferon is being produced. So that starts to get pretty complex pretty quickly. So if this is happening at a large scale, could there be some way that production of interferon is helping a human kind of push back on viruses that are trying to infect the human body? And could this production of interferon ever be coming through phage interactions with human cells? I don't know the extent that that would be occurring. I don't think the extent would be a great one, but I'm saying that biologically that's possible. So the second part of the question, I've forgotten what it was. Sorry.
Yeah, the first was you answered. I think the once you said that you haven't observed any interaction between bacteriophages and any kind of viruses, and the second is the immune response anticipated.
Oh, right. Okay. Thank you. So so remember, phage particles are the same size, generally in many cases, as viruses that would infect eukaryotes, including those that would infect humans. And here my point is, the adaptive immune response, I think, is the more critical one here. So if the adaptive immunity of humans is seeing a foreign invader, recognizing it, producing antibodies against it that are going to be remembered through time and produced, then that would be the response. That could explain how you deploy a phage at very high density over a period of time in a human, and then eventually this adaptive immunity builds up because it's such a high level invader. And in that human, this is remembered through the adaptive immune system, and you try and deploy the phage again, and you can't do it because of adaptive immunity. So that is plausible. I'm not going to say that that couldn't happen. Anecdotally, we haven't seen it. We've had some people who we've delivered a phage to through emergency therapy, and we've waited a multi-month period, and they've had some lingering bacterial load that we want to try and push it down further using the same phage. And when we deploy it, we find that the patient improves. So that, again, anecdotally, and we're looking for more confirmation through analyzing samples over time in these types of patients, that it suggests, at least in that case for that phage in humans, it wasn't a problem, because that was easily enough time for the adaptive immunity to kick in and potentially for that phage to be seen as an invader. It was not targeted by the immune system, at least didn't seem to be, and we got a good outcome. So I wouldn't say that's happening all the time, but that certainly is something that we need to keep looking at.
So Anisha Adhikari, I hope you got your answer. Ali Jensen wants to know that most phage therapy is currently personalized. Can you see broad host range phage therapy emerging? And if so, then what kind of phage cocktails would have to be made? Phage mixes need updating. So yeah, broadly she wants to know how will it be a broad host range phage therapy emerging?
Golden question. So I think there are a lot of different ideas about this, but there's no absolute, and it could actually be a different strategy depending on what the disease is you're targeting. So if you found a very generalist phage that covers a lot of genotype space in the target bacteria, then you might be able to generally use it alone. You might be able to, on the other hand, bundle together phages that are quite specialized in what they are doing for interactions with the bacteria, put them in a cocktail and use that instead. Or another example would be deploy one phage and then stop deploying that one and move in sequence to a different phage. And the reason you would do that is to sort of push back on some problems in the human, like maybe reducing inflammation at the site of the infection. We've been able to kind of find phages that would essentially be anti-inflammatory in the lungs. And then you kind of move on to, okay, now I'm going to push against antibiotic resistance in that population of bacteria and try and make them more sensitive. And you take sort of the staged approach, because medicine and the physicians are thinking, I need to consider one problem first, get it under control, and then move to the next one. So with all these different strategies and possibilities, again, follows a lot of basic research needs to be done, especially on the evolution. If you are using one strategy versus another one, I, as an evolutionary biologist, would care, is one of those strategies more predictable for the thing that happens next in the bacterial population relative to other strategies? I think that that's really, in my opinion, the heart of the matter. We want to deploy a therapy and know what happens next in terms of the bacterial potential resistance to that therapy. And the ability to do that accurately arguably could be, you know, as simple as you can deploy the therapy, the more accurate you can generate the predicted next step. So that's kind of what we favor, is keep things as simple as possible. Try and use a generalist phage, try and use phages in sequence. But I'm not saying in other circumstances and in the hands of other individuals that they might show other strategies might be reasonable ones to address as well.
Right. Several comments, Professor, saying exciting talk and indeed a great talk. So people are really enjoying. This question is, I think it is somewhat covered in your lecture, but still there's a question. If a bacterium becomes resistant to phage, then whether the bacterium also loses their virulence to their host. So any other example probably Dr. Mahananda is trying to ask besides.
Right. Yeah, I didn't touch on that too much today because in the interest of time, but yeah, there's some systems where we've seen bacterial pathogens that have proteins that let them, for example, move intracellularly, or between, I should say, intercellularly. So if they are bacteria that invade your cells to make you sick, that can often happen in the gastrointestinal system. Shigella would be an example. So if you find a phage that exploits these bacteria by binding to the exact proteins that let the bacteria move from cell to cell, then they're going to select against those proteins and prevent that bacterium from being an efficient intracellular pathogen. So the problem is, how are you going to reach those bacteria once a person is already infected? And here I'll quickly say that there are lots of ways in vitro to show and discover phages that look promising in therapy, and then you need to grapple with how do you deliver them. And if you are going after a bacterium that is within your cells already making you sick, then you have to have a delivery mechanism that gets the phages inside of your human cells. Even if they're entering at a low rate, it's probably not at a high enough rate to be good for you. So the idea there is wrap the phages in something that is recognized by the cell as being okay for uptake. So you could wrap them in a liposome or something like this, use nanotechnology to your advantage so that the cells in your body see, oh, that's the thing that's okay for me to ingest. And unknowingly, what they're doing is taking in the phage, and then that is able to see the bacteria inside of the cells, and it'll do, hopefully, the job of attacking those bacteria, killing them, selecting against the ability of the bacteria to move from cell to cell. And I went through a kind of an elaborate scenario there to illustrate how even the hard to reach pathogens, bacterial pathogens, might be subject to phage therapy if you can be creative enough about the delivery.
Okay. So Dr. Aradhana from Vipra wants to ask again. She says, thanks for a wonderful talk. She wants to know, how did you arrive at the quality parameters like the endotoxin levels which are permissible for phage uses in clinical trial?
Yes, very good question. So essentially, for our emergency treatment, we put the phage prep through a dialysis procedure that eliminates endotoxins, and the purity of that could be tested by laboratories that are not my own, and they take the data and they talk directly with the U.S. Food and Drug Administration to verify independently that those preps are fine. That's essentially the path that we've taken for emergency phage therapy, we and others. And that needs to be done at scale if you are producing a large number of doses for, say, a clinical trial. So that's where essentially what I just described would be done through bulk manufacturing of phages in a laboratory that was capable of doing that at scale. And then they're doing the testing and also reporting to the US FDA, in our case, that indeed those preps, those doses are fine. There's no debris in there that could be harmful to humans. And that's essentially a parallel analogous process that could be done in a laboratory at small scale and verified, or it could be done by a manufacturer at large scale and also verified.
Two questions for plant phages in plant system. Dr. Mahananda wants to know that, is it possible that we apply phages directly in agricultural fields without genome characterization? Can they be used if they show the activity against the pathogen? And the other question is by Porti, sorry. She says, thank you, Dr. Paul. About the implication of receptor in genomics for phage therapy, she wants to know that this ecological binding is also interesting for biocontrol agriculture. So can this be used in plant pathology as well?
Yes. So the example I gave where I described to the audience, I've been kind of thinking about this stuff for a while because I first observed it in plant pathogenic bacteria that we happen to be using in the laboratory to grow an RNA phage called phi6. So whether the regulatory hurdles, for example, would be easier in a plant system to get over those hurdles. And I think the question was sort of, could you use a less characterized phage in those circumstances? It all depends in the particular country, what the regulatory hurdles are and what the expectation is for your understanding of that system. So I just have to be honest. I assume that in plant agriculture, for example, probably the regulatory hurdles would be a bit easier to step over than they would be in a human system or even a domesticated animal one. So therefore, I have a lot of confidence that you could do things properly and maybe not as in-depth characterized potentially, but it really depends on the rules and regulations of a country. But in a sense, the second question, I have a lot of optimism in plant and animal agriculture. And that's because necessarily this stuff is harder to do in humans, as you would expect. The regulatory hurdles, the barriers are more difficult. And if you're seeing these successes to date, and if we start to see more successes from clinical trials, then I think sure that we should expect in plant and animal agriculture and aquaculture, let's try as carefully as we can. I think the way to go is collaboratively. If you have people that start to really become more expert in some subfields of phage therapy, this is naturally going to occur. So many people are entering the field, a lot of young investigators, and they'll become probably a little more specialized for certain aspects of it, but let's try and work together against these problems. I'm especially worried about, with climate change and other things that are already threatening the sustainability of arable land. You have to think about food security being affected by antibiotic resistance spreading in plant and animal agriculture. So these are really big issues, and anything that we could do creatively to push back and help solve them, I think, is worth considering.
Absolutely. Gokul Nair, please indicate, because there's so many more questions. That's about how people are excited with your talk. So I think I can take a few more. Is that okay?
Yeah, maybe we could take out. Unfortunately, like everybody, I have for my work day, it's just starting. So maybe another question or two, and then.
Okay, so my apologies to those who all questions can't be taken, but there are several very interesting questions. Some questions are also on endolysis. If you would like to take, otherwise I would. There are a couple of questions on use of endolysis in treatment of anti-resistant infection. So if you want to take that, otherwise I can take other questions.
Sure. Let me. I'll try that one. So yeah, I'm very excited about people who are trying to develop. Can you take things that are produced during the replication process of phages and just harness them to try and use that against the bacterial population in therapy? And could those types of mechanisms, if they're bundled together with lytic phages, do an even better job than either of them alone through synergy? I think these are plausible outcomes that need to be examined. So I'm quite excited about that. And I think in the probably not too distant future, we'll try and collaborate with people who are trying to do this, because it is worth considering. If I could just say for a moment, that kind of technology is, of course, more similar to chemical antibiotics in the sense that you're dealing with something that can't evolve on you. It can't change. A phage can change, whereas an endolysin won't. So you can sort of make it in an amount and deploy it, and it won't change on you. So it's quite exciting to think about different technologies coming together and synergizing.
So one more question, Raju Mukherjee he's joined us. So Raju, do you want to ask directly, or should I read your question?
Oh, sure, I can ask directly. I just wanted to know, if we consider this interaction and analogous to a host pathogen interaction, is there a heterogeneity in terms of outcome of this interaction? And if you have mapped any such, loci at either site, and if that can be helped, to make a superlytic phage, for example.
Interesting. So by host interaction, you mean the bacterial host interacting with the phage? Yeah. So I think that's a great question. In a sense, what we and others are starting to do is look at the so-called co-evolutionary treadmill between the phages and the bacteria. So as you are deploying the phage, you're going to think that what I've been talking about is the evolution of phage resistance will happen on the bacterial side, but you also have the ability of phages to evolve to overcome that resistance. And you can have this counterattack happening. So if you examine that in the laboratory over time, that's multi-generational, that could be happening in the patient, and that gives you insight into, all right, some of these genetic changes could be scattered around the bacterial population, important in resistance, but what's really happening in that bacterial population over the longer haul? That's what you have to be more interested in, because it's going to be more important. The early changes that a bacterial population could do to push back on the phage, what is happening when the phage itself pushes back and might be useful in therapy going forward with coevolution. So I've spent a lot of time today talking about the kind of first mutations that occur in a bacterial population and predicting what those are, it's a more difficult thing, but certainly plausible to examine. What are the more long-term downstream consequences? What are the co-evolutionary rules? And that kind of your question reminds me of that realm, is can you, and I guess it's my last opportunity to bring up another favorite thing, is evolvability. Can you predict over many generations and time how systems will evolve? And I think that that's where this kind of technology might be useful.
Sure. Thank you.
Thank you.
Great. Several more questions unanswered, but maybe it's for some other time, right? Because you have, right? or if you can take, is that okay? Should I, or should we close it now?
So I'll have to go to, unfortunately, my next meeting soon. But if there's any way for you to send some questions my way, or that would be wonderful. I may not be able to answer every last question if people reach out to me by email, but
so what we'll
thank
do
you so
is,
much for your interest.
I will take this, I will send you these questions, and if time permits, you could send the answers, and we would send them back to the, that would be wonderful. Wonderful.
That sounds great.
Thank you so much. So Konanika, are you there? I mean, for the formal vote of thanks. So thank you very much. I'm so happy.
You're quite welcome. Thank you.
And your talk and the wonderful question answer session. I'm very happy with this entire evening. Thank you so much.
Thank you so much.
Yeah, hello there. So I'm going to keep this very short. Thank you very much, Professor Turner, for a very exciting and stimulating talk. And this was also reflected by the number of questions that this talk has generated.
I'm delighted.
In fact, the International Bacteriophage Research Consortium was created with this in mind, to address the challenges by bringing different scientists with different expertise together to address the challenges that are standing in the way of bringing phage therapy into a more widely accepted form of therapy.
Fantastic. It's definitely the way we need to go.
Yes, you have elucidated the biological mechanism and the biological challenges very lucidly. And then, of course, going on to manufacturing and regulatory hurdles that we need to overcome.
Thank you so much.
I hope that we will continue to interact and work together in the future.
The same here. Best to you and the others. You're doing tremendous work. I think we could do even better things together. So thank you for your time and attention today. And I really appreciate the invitation.
Thank you.
Thanks for all the,
sorry for me. I'd also like to thank the audience for being here across different time zones, difficult time zones in some parts. And unfortunately, all your questions couldn't be taken, but that has made it a very meaningful talk. A talk without questions would be, I mean, the talk in itself is very interesting, but the generation of ideas is what is also very important through the questions. So hopefully these questions will be answered over time. And also, although last but not the least, as I say, the students and volunteers working in the background, Ritu Arora and her team from ANDC, and Kopal Sharma and her team from OHSL. Thank you very much for making this a smooth talk. And thank you, everyone.
Thank you.
Thank you, everybody. Have a very good day.
You too.
Bye-bye.
Thank you. Thank you all for joining. Great. So, Konanika, we close this. That was nice.
Are we still live?
Oh, yeah, we must be live. Yeah. So